Starting the stream. Starting the stream.
Check, check.
My gosh, that's loud. I cannot just. Oh yeah, I can just turn down the volume of this. Okay, let's do this. There we go. Double check. Let's go.
Check, check.
Nice. That's. I think that's a good volume. Okay. Offline chat. Okay, okay. Let me just double check. Okay, okay. Okay, everything seems fine. Everything seems working. Let me double check. It is. Okay, I hope this is recording. So I can actually use it for my project. I don't know what do I do. Okay. Let me just open this. Hello everybody. Welcome to my oyster stream. This is just something I'm gonna do for my video project. Just kind of make things a bit different, make things a bit. Some of my teachers actually give me extra marks, and it's not just the same videos over and over again. Yeah, what I'm gonna do today is I'm just gonna explain everybody what I did for so many weeks with my oysters, because I think all my friends have heard me say, I'm too busy, I can't go out this weekend, I have to take care of my oysters and stuff like that. So what did I actually do this whole time? And yeah, so that's what this stream's gonna be all about. And I highly suggest everybody in chat to just chat in the chat box. So because I have this chat to speech thing open, let me make sure that my speakers are on. Yep. So everybody can hear the chat, the voice when you type a message in the chat. Can make things more fun, more interactive. I am broadcasting this live, so Dr. James, I hope this is more interesting for you to mark. Yeah. So let's start with basically why the heck are we doing our project on oysters? So there is two major reasons we actually started doing this. The first reason is because there is actually a growing demand for like shellfish and like sustainability in Singapore, right? And Singapore actually has this 20 by 20 30 by 30 thing, sorry, wrong number. 30 by 30 campaign by the Singapore Food Agency, where they want to produce like 30% of Singapore's food inside Singapore by 2030. So that's 30% of our food supply by 2030 grown in Singapore. So we don't have to like rely on other countries and stuff like that, because imagine if there's like touch wood, something bad happens lah, not gonna have any food to eat, right? Yeah, so that's basically why we are doing this project. It's kind of help Singapore sustainability a bit more, and yeah, so that's basically it. So the second thing is just to help our local oyster farmers, because they have noticed that during periods of high rainfall, right? So let me just open this up. So imagine this is the sea, and this is like our oyster farms. Pardon my drawing. And then during periods of high rainfall, they have realized that for some reason oyster mortality, or like the rate at which oysters die, increased so much during this time, and we don't know why. We have no idea why. So we are doing this experiment to like kind of see what about the rain actually like affects it, or if the rain actually affects it at all. Maybe something other than the rain is contributing. So really the first things we think about, right, when we hear about this high rainfall causing mortality, imagine it's the oysters. Yeah, pardon my drawing. Yeah, so the first thing to think about, right, when it rains, rain is fresh water, right? Yeah, and seawater is salty. So if fresh water mixes with seawater, especially in the case of rain, which is like in a local area, that it's not like throughout the whole world, right? It's just a region. So if fresh water mixes with seawater, it makes the water less salty. So we call this salinity. The salinity actually drops in the water in the area. So one thing we are testing in our test is actually to see whether the lower salinity will actually like make oysters more stressed out. And because stress is such an important thing in farming, because if the animals are stressed, they die more easily. Yeah, especially in stuff like fish, oysters, seafood. That is very important, managing stress. So we want to make sure the environment is as comfortable for them as possible. And yeah, so the lower salinity might actually contribute to increased stress and increasing the amount of deaths in the oysters. Then the other thing that is a bit more obvious for everybody, I'm sure y'all could figure it out also, is that rainfall is generally drops the temperature of the environment. So the temperature may drop and cause my gosh, yeah, so the temperature may drop and does the same thing as lower salinity, where it changes the water temperature and then the oysters don't like it and they die from the stress it creates. So that's the background of what we are doing for what I did during my oyster project. And basically our aim here is to kind of see, is to judge the effect of oysters. Hold on, let me pull this up. Yeah, so it's this please here, please let me move, there we go. Yeah, so it's to see what the effect of like different salinity and different temperature levels, like how does it affect, because the mortality, first of all, the how many survive if we change the temperature and salinity, and then how much they eat, and also how does it affect the gonads, or what we call the reproductive organs, as you may know. But why do we need to care about the reproductive organs? That links back to my first part where we were talking about sustainability. If we are able to get like better reproductive organs in our oysters, right, we are able to have them reproduce in Singapore instead of having to import the baby oysters from overseas and then we grow them here, because that's exactly what we're doing. Yeah, so if we import the baby oysters, we are not sustainable by ourselves, and if we have better reproductive organs in our oysters, we can actually try reproducing in Singapore and actually be more sustainable and farm the oysters from start to finish all by ourselves. Yeah, so that is basically the background of what we are doing today. Let me just erase this monstrosity. Yeah, so next thing, we're gonna talk about what the heck did we even do with our experiment, like what did we do. Basically what we call our experiment setup. Yeah, so this is basically what we did. We had 12 tanks full of oysters. Each tank had 10 oysters inside. Okay, so this is about to get complicated, so let me draw it out for y'all real quick. So first thing first, we have 12 tanks, right? But the 12 tanks don't actually have different stuff. We call these treatments. So we have 1, 2, 3. Actually we have 4 treatments, but the fourth one is called a control, which is normal setting, the normal setting. So this will be the control. So the control will have high temperature, because Singapore is hot, right? Singapore is around 30 degrees, so the high temperature is around 30 degrees, and it has high salinity, which is around 30 parts per million, parts per trillion. Yeah, that's the unit we use to measure concentration of the salt in the water. Yeah, so this 30 degrees, this is to replicate what Singapore's SG's situation, or what's the better word for it? SG's think. Let's just say situation for now. Yeah. So for the first one, right, we want to see what happens if we just, we drop the temperature but everything else is the same. So if low temperature, low temperature, we call this ambient temperature, but for now I'm going to say low temperature. So the low temperature is around 22 degrees, so it's a major drop compared to what the 30 degrees is. But we're gonna have the same high salinity, high cell, and this is to have the replicate drop in temperature. And then we're gonna have blah, 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 blah. We're just going to drop the salinity, so the temperature is still gonna remain high like Singapore, but the salinity is gonna be low. Around 16. So it's around half. Half fresh water, half seawater. And then the last one is going to be the most chaotic one, where we see what happens if we drop the temperature and the salinity. So drop temp, no, what am I talking about? Low temp, low salinity. Yep. This is basically our setup. And you may be wondering, we actually had 12 tanks beforehand, right? So why are there only 4 here? Well, you see, when we do experiments, we actually have to, we have to find like averages and stuff, right? So that it's more reliable. Because, anything can happen to that one tank and we might not know if it's due to what we changed or whether it's something else. So each of these tanks will have 3. So, yeah. Ish. No. So this gonna have 3. This gonna have 3. And this gonna have 3. So altogether it's gonna be 12 tanks that we have. And that's what you see over here. Yeah, so that is basically what that is. Every single day we change 30% of the water to like clean the tanks and stuff. Oh yeah, before that, in each tank actually, let me just oh my gosh, no. So in one tank, one tank is gonna have this setup, okay? It's gonna have the oysters. We're gonna have 10 oysters. Oysters times 10. Can you even understand what I'm saying? What I'm writing? Hold on. So this is an oyster, okay? Then we're gonna have, for those that are higher temperature, right? We have one water heater. This is the water heater. This is the wiring connected. It's electrical. But it won't electrocute the water because it's waterproof. Waterproof. And then we have one sponge filter. So you might have seen this in like normal fish tanks in like fish shops and stuff like that. So a sponge filter looks like this. Boom, this right here. So we have this. Inside the sponge filter actually has grows a lot of bacteria that is very useful in our fish tank, and it does like the nitrogen cycle. You can research more if you want to Google what that means. But yeah, that's basically the setup of what our thing is about. And then, yeah. So basically, to end off the first part of this topic, to end off the first part of this topic, what we actually, oh my gosh, there we go. So what we actually hypothesize, right, or what we guess happens is that the low salinity and low temperature ones will have the most deaths. That's what we guess, because as we've observed before, rainfall creates these conditions. Conditions, that's the word, not situation. Conditions. Yeah, creates these conditions where it drops the salinity, drops the temperature, and yeah. So we hypothesize that these conditions is the worst for oysters growing in Singapore, which will lead to increased death, which we observe in the farms. So that is the end of the first part of my project. Now we move on to stuff that gets okay. Let me double check. Let me see. For this part. Okay, I don't think I can do this in stream because I haven't organized all the videos. Let me just double check the videos real quick actually. While we're here. By the way, if like anybody new is watching the stream right now, feel free to chat in the chat box. This website will actually read out your messages to me and I can actually respond. If you have any questions so far about what the thing is about. Yeah, so we actually have so many videos. But yeah, I don't think I'll go through what we did, what we actually found in our thing. Just like some highlights we have. Oh wait, it's probably not here. Never mind, no highlights. If you want to see the highlights, you're gonna have to watch the video I'll post on the YouTube channel. Yeah. So just because you're here doesn't mean you get everything, okay? So next, we talk about the exciting part. Let's talk about the results. Let's go. So let's just spend like a good 2 minutes per result, so we can actually like see what happens. Yeah. So this is actually our poster, by the way, if you don't know. This is what we had to present to our teachers. It's a lot of information for one thing. Okay. First thing on our list, let's talk about. So let's talk about what we found in our oysters. I won't go through what. Wait, hold on. Let me actually double check my. Sorry, it's a bit chaotic. Discussions and takeaways. Yeah. 1 second. Okay, sorry. The reason I'm so quiet right now is because I've realized that I've overlapped a bunch of stuff, and I'm trying to figure out how to break this up. How to break this up between videos. Results discussion. Okay, you know what? Who cares? It's just AQ. It's just a module. It's gonna be over by this Sunday anyway. Yeah. So let's just go through what the results actually are. Okay. So let's start with weight and length. So ambient. Just so everybody is clear, yeah. Ambient temperature. Ambient just means the normal conditions inside the room we had, or like the normal things without any changes, that man-made changes. So for example, the ambient temperature inside the room, right, because the room is air conditioned, the ambient temperature is 23 degrees.
1, 2, throw.
1, 2, 3. Okay. Okay, so yeah, ambient temperature just means low, ambient salinity just means high. Okay, just remember that. It's basically the opposite of low and high temperature. Yeah, so just remember that. I'll just explain what they are as we go along lah. So ambient temperature and ambient PPT, which is the salinity. Ambient temperature is low, so low temperature and high salinity gave us 3.9. Oh no, sorry. High temperature and low salinity gave us the longest or like biggest oysters, which is 3.9 centimeters, almost 4 centimeters. Pretty much 4 centimeters. Then it's followed by low temperature, high salinity, and then after that it's low temperature, low salinity at 3.8. Then the lowest is actually high temperature and ambient and high salinity, so it's high. And actually, what's the most interesting about this, right? If you notice, this is actually high temperature and high salinity is actually the conditions that Singapore is growing our oysters in. So if you see the weight, it also gives us the lightest oysters. So like they're not feeding properly, they're not growing as well. So we're starting to think maybe our hypothesis is something has something wrong. Maybe Singapore's conditions aren't the best for our oysters, and that's why they're dying. Yeah, so that is something to think about. And then, okay, now we move on to, so this is basically the salinity, the average salinity and the average temperature. Nothing too big here. And then now we're going to talk about ammonia and nitrite. So I guess those of you who are not in the science scene, right, or like actually a lot of you who don't even like take care of fish don't really know about ammonia and nitrites effect on sea life. So actually this is gonna be one of our biggest like things we look out for when we take care of sea animals, because ammonia comes when you like when stuff decompose in water, when like you know uneaten food decompose and then it like the bacteria yeah, like stuff just decomposes in the water, like food, dead animals, and like especially feces or like shit, yeah. So if they start breaking down in the water, they release ammonia, which is very, very unhealthy for the fish and may actually die. It's one of the most important things you look out for. So throughout this, we actually should have made a graph lah, but we just put numbers here, so you don't really know what you're seeing. But basically, we want to keep the ammonia level below 1.0. So as you can see, they're all below 1.0. The highest being like 0.65. So it warrants water change, but it doesn't warrant like a full tank of water change. This is only like 30%. So we change 30% of the water and stuff like that. The nitrite is similar. It's in a similar thing. So one of the things that we have to take note here is that we had brand new sponge filters that didn't have the bacteria inside. So it had to take a while for bacteria to cultivate. So that's why some of these go quite high, 0.65, 0.64, and they are not stable at like 0 and stuff like that. So it's a cycle lah. It also cycles high to low, high to low. So that's why there is differing numbers there. Now on to a bit more interesting stuff. We find bacteria. Indoor test is to find enterobacteria inside our oyster samples. So we actually kill the oysters and then test on them lah. If you are not aware, rip oysters, I guess. Yeah. So indoor test is to test for enterobacteria. So if there's an absence of ring, that means there's no bacteria. If there's presence of a ring, there is bacteria. So it shows that high temperature and high salinity has the bacteria, and high temperature, low salinity has the bacteria.
I'm definitely not aware.
Well, now you know the oysters die. I killed all of them. Okay. Yeah, so the indoor test to test for bacteria showed that the high temperature ones actually had bacteria inside. And it actually makes sense lah. Bacteria grows well in higher temperatures. That's why we keep our food in the fridge, right? So it keeps it cold, and so bacteria doesn't grow and like proliferate inside in our food. And yeah, as you can tell, the same test, like a different variation of the same test, shows all negative. So what we think is, because this test is less sensitive.
How dare you kill those oysters and all those bacterias?
No. You can blame my teacher. I don't know. It was necessary. Okay, it's for the sustainability of Singapore. It's for your own good. Yeah. So as I was saying, this indoor test is all negative, but this one shows some positive, and this one is a bit more sensitive. No, this one is a bit more sensitive. So that's why this one might have false negatives, and this one is more sensitive, so it might have. So it probably able to find the bacteria even though it's smaller quantity. Yeah, so if you can see the bacteria count here, we actually like made the plates. I can actually show you the plates over here. Come on, show me. Yeah. So this is what this is. Let me see how dare you but. So this is what our plates look like. As you can see, we got some like disgusting plates here. This one is like full of bacteria. Yeah, so each
WO.
yeah, so each circle actually represents one bacteria. So one colony like multiplies into one circle basically, so that we are able to see. One bacteria multiplies to one colony basically. That's how we count the bacteria in our samples. Not the most how to say reliable, but it does the job. So this is the numbers we have. We had two duplicates, so that's why it looks like coordinates, but it's not. It's just the first duplicate 106, second is 97. Yeah, so as you can see, the high temperature ones have very high numbers, and the low temperature ones, especially the low temperature and the high salinity, for whatever reason, has the best is basically the cleanest. And why is that? Is because salinity, some bacteria can survive in low amounts of like salinity, but when the salt water goes up, less freshwater bacteria are able to like grow inside the water, so that's why there's less. So that's why the high seawater low temperature one has like the least amount of bacteria, and then the one that is slightly fresher water but still low temperature has like moderate amounts of bacteria, but the high temperature ones just like 100 plus. It's crazy. Yeah, so basically this is just to show the feeding rate, so how much they ate over time. Yeah, so this 30 minutes, 60 minutes, 90 minutes, 120 minutes. Yeah, we just basically how this happened, we fed the oysters, then we waited, and then we counted the amounts of algae cells inside the water. We just take a dropper and then we count it under a microscope. So this 75 is not 75 cells, it's actually 750,000 is times 10 to the power of 4 in just 1 ml of water. Yeah, so if you think about it more, it's a lot. The tank is like 40 liters, so it's a lot of cells inside lah that we're not able to count. So as you can see, this graph doesn't look exactly like this graph. Like over time you expect the amount of cells to go down, right?
Much.
What? Yeah, exactly. Thank you, Shanoa. So as you can tell, right, this graph looks a bit wonky. It's supposed to go down like this lah. As time goes on, as the oysters eat, it's gonna be less cells, right? But what you actually have to take into account here is that we had to reuse the glass slides because the plastic slides under the microscope because we didn't want to waste so much. We had 12 tanks and we had to do it like 4 times, so that's like 24 times 48 slides that we are going to waste. So yeah, we basically just like reuse the slides so we don't hurt the environment. So this is colorimetric test. The colorimetric test you can see over here. Yeah, the thing that was there for a split second, where was it? Yeah, this thing here. So as you can see, right, it's just like this device that will actually
Are you ready?
yeah, it shines a light on the oysters, and then it actually like takes the like the RGB, but it's like this LAB or whatever. Yeah, so it's kind of like to see whether the color of the oysters in the different treatments are different from the color from the normal treatment, which is the high temperature, high salinity, the Singapore's conditions. And as you can see, this is the Singapore one. So these are colors. These all fail except this one for some reason. We do not know why. Yeah, it is unknown why the high temperature. One hypothesis, like one guess lah, I have is that the standard sample might not have been the best sample to use also, because we actually killed the oysters the day before. Yeah, so and put in the fridge, so the color should have most probably like changed. and that is why. Now we move on to mercury test. We're going to speed up a bit because we're running out of time. So for mercury test, you can tell that the ambient temperature and the low the low temperature high salinity had low mercury inside, and then the high temperature low salinity also had low. This one also low, but this one is so high. But why is it so high? One thing we guessed is that because these oysters were taken from the Straits of Johor, right? Some of these oysters may have taken the mercury found inside the water, because it's a naturally found metallic element, so it could have been found in the water. But what we kind of guess more likely happened is that this was due to human error, because the sticks, like the test we use, which is called dipstick test, actually it's just like the color looks a bit off from the chart that we had, so it's anyone's guess really. But when we saw this test, it kind of looked like the same as the rest, but our teammate recorded it as 50, like according to her vision lah, and we just put it in. But yeah, just know that the mercury test most likely due to human error. Now is the filth analysis. So basically we just like find for any like physical particles inside the oyster samples that we have. As you can see over here, we found like some like brown stuff inside the oyster. We found some hair. You can't really tell, but there's like some algae found here, which you don't really want to eat. Like, it tastes a bit gross. It smells like seaweed. Every time I feed the oysters, I want to vomit. Yeah, so it's not the cleanest oysters, most probably because we didn't really focus on cleanliness. We weren't really planning on eating them. Yeah, so that is probably why the filth analysis looks like that. And lastly, we check gonadal development. This is important. Gonadal development, if you forgot, is basically the reproductive organs, how much they've developed and whether they even developed at all. So each one, we killed 8 oysters each from each treatment, except this one we had 7 for some reason. Maybe that wasn't enough, I'm not sure. So the numbers with gonads or reproductive organs is only like 3, 2, and this one only had 1. So this one is a fail. Had no females. This one also no females. These two fail like. So basically, high temperature not really the best for reproduction in our oysters lah. Yeah, and then really the best is for lower temperature, best for reproduction. And they really just like have similar results, not really much difference. So from this we just take into account high temperature bad for oyster babies, yeah, or oyster woohoo. So this is the eggs under microscope. They're microscopic. Yeah, that's why you've never seen an oyster egg. And then this is oyster sperm. Each dot here is 1 sperm, and these are the eggs, so big and so small, can't even tell from here. And now for our favorite, mortality. So how many died in each one? So contrary to what we believed at the beginning, the low temperature, low salinity had the least amount of mortality, which is weird because this is exactly the conditions we had from rainfall, which we believe was what caused so much deaths in the farms at the first place. So we are scratching our head. Maybe, yeah. It's weird. Yeah, so this like really bamboozled us. We were so confused. Why is the cold water and fresher water killing the least amount of oysters? And we have no idea. So what we can conclude basically from this is maybe the rain isn't what's killing them. Maybe it's surface runoff, stuff. The rainwater from the surface go to the sea and make the sea dirtier, cloudier, or whatever. We don't know. That's for another experiment, for another day, for another group of students to do another project. That's not my problem. Yeah, so the highest one actually is high temperature and low salinity, and then right close behind is the high temperature and normal salinity with the high salinity. This is the Singapore condition one, and we can tell from this and also the weight of the oysters, maybe Singapore not the best to farm oysters, or at least don't do it, like try to change something about Singapore's stuff. Yeah, and that is all for the results of our oysters. Yeah, that was a lot of numbers and stuff. I hope you all understand. If you all have any questions, just ask me in the chat below. Let me just bring up my our document that we used for our what do we call it? I can't remember. Shellfish? No. Project. No. I'll just put doc and then find it there. Ayo, is it this one?
So we eat oysters elsewhere.
I don't know. I mean, not really. We don't have to eat the oysters from somewhere else. We just need to find a better way to farm them, because Singapore is just not it, fam. It's just not it. Yeah, Singapore's water's not the best for oysters. Where is it? Come on. There we go. So what can we do to help farmers in Singapore? So there is a few things that we can do. Like to meet the higher goal, right? We can somehow wait, hold on. Let me bring up the conclusion here, which is where I wrote this. But yeah, I don't have to read it. So what we have to do is somehow in the oyster farms in Singapore lower the temperature and lower the salinity of the seawater that is being farmed in. Because if you all don't know what oyster farms look like, they look like this, okay? Yeah, they look something like this. They're just on the on the surface of the sea, and just like that in the normal waters. So how the heck do we like change the temperature and the saltiness of our seawater? And there's not much we can do, but okay, so for the high temperature, right, we actually can do something about it. If we are able to farm oysters in a deeper depth of water.
I'm
right?
sorry I'm late, prof.
Don't worry, Teha. We just reached the final part of our thing where we talk about what farmers can do. So for a high temperature, right, how can we combat that without actually changing the temperature of the water? As we know, the oysters grow better and like can reproduce better in like lower temperatures, right? So if we farm oysters in a lower depth from basically the freaking surface of the water here, maybe the water will be colder because it's further away from the sun. The most of the water, some of the heat from the sun is actually reflected back, and yeah, water is more of an insulator of heat than inland areas. So the deeper the oysters are, the cooler the water will be. I'm sure you all have realized that when you go to the seaside or when you go to the beach and the water is actually colder than the air, like the area around it. Yeah, so that is one thing we can do for temperature. For salinity, there's really not much we can do lah, unless we just have like yeah. Wait, hold on. Like one idea I had was like, imagine it's the sea and this is the farm, right? We have like pipelines going from the mainland Singapore to the sea, and then it's just like showering. No, what am I doing? It's like showering the oysters in
Where
fresh
to find
water.
colder water?
If you go deeper under seawater, right, to find where to find cold water. If you go deeper, like water naturally gets colder the deeper you go in the lower the depth. Yeah, so you can, if you are able to farm oysters, like just lower this down, man. It's not that hard, just lower it a bit. But one thing about Singapore, right, that's a bit hard to do because Singapore is so shallow, our seas are so shallow. So that's one of the problems we have with these farms. Yeah, so um where was I? Oh yes. So for salinity, right, it's a bit different. We can't exactly lower it to have lower salinity because seawater is like salty everywhere. Yeah, so if we shower the oysters in fresh water, it will change the salinity of the water and
Hello.
make it less salty. Yeah, it is shallow. Yeah, so if you shower it, it'll be less salty, but oysters, but this is a bit not sustainable. We're gonna run out of water in Singapore and we're gonna be SG sad, okay? We're gonna have no water, we can't shower because the oysters are showering. So that is probably not the best way to do this. And that is why my genius brain has come up with this idea. Ta-da! Okay, y'all may be looking at this like, what the heck is this? Okay, this is a semi-closed farm for salmon. Okay, so what does semi-closed mean? So an open farm is basically what we see here. Basically, the oysters are just farmed in the open sea, just inside like baskets to like, you know, so that they are not everywhere. But a semi-closed farm contains them inside.
Menstrual cup, I'm so sorry.
Excuse me?
Why it look like a huge menstrual cup?
It's not a cup. It's not a menstrual cup, okay? So I like how you both had the same thought. It's not a menstrual. Okay, fine. It's a giant menstrual cup in the middle of the sea, where people farm salmon. But what if we use this thing to farm oysters instead? Okay, hear me out. Okay, hear me out. This thing here separates the oysters from the outside of the sea. So if we are able to pump fresh water, because these things here, right, you see these things here, these like protrusions at the bottom, they are actually
It's a menstrual cup for my fishy bits.
enough please okay, as I was saying, I'm gonna ignore that. As I was saying, these protrusions actually pump seawater, and because they're lower in depth, right, they actually pump the seawater from below, which is colder, into the farm. So the farm water actually will be colder than the water outside, like over here. So that is something cool about this. Maybe I can copy and paste it here. Let's go! Yep. Now I can draw. Yeah, so these things will suck in colder water from this area and it will bring it inside the farm here. And then what we can do, right, is like half of
So
this.
it's like a scrotum but for colder temps.
Hey, enough this is gonna be my project video Yeah, so half of these pipes, right, can suck in fresh water and half of this can suck in, oops, what happened to my pencil? Come on, man. No, I cannot draw. Not, yeah. And half of this can suck in seawater, so half fresh, half sea. This will lower the let me just erase. So this will make the temperature inside here drop. Temperature. And it will also drop the salinity without having to like constantly have seawater, fresh water going inside. You know, like the previous showering of the oyster idea. Yeah, so the fresh water will be contained inside and we'll use much less. Still a lot, but much less. And we'll have a controlled area inside here to control whatever we need. So if we need to drop the temperature, we can drop the temperatures one way or another. And if we need to drop increase salt water, for example, you can just add salt or whatever, or add more fresh water if it needs to be fresher. So this is something that I've actually this is more of like a this is an idea I have for farmers lah, but obviously the thing is it's gonna be very expensive. Yeah, so that is one of the things why that is one of the reasons why this idea might not be the best, but it's an idea nonetheless. Hey, that rhyme, let's go.
So can control the temperature and water, just confirming.
Yeah, you can. So it will collect cold water from the deeper area here to make the inside colder, and then we can control the salinity.
So is there a cheaper alternative?
Cheaper alternative? Smaller farms, I guess? Or maybe we can have like maybe instead of this we can have completely closed oyster farms. I don't know if that's a thing though. Closed oyster farms. Yeah, I don't think that's a thing. So what closed farms mean is basically it's just completely separate from the wild, like the natural habitat. So we put in our own water, we put in our own salt and control everything there. I'm not really sure whether it's more expensive than having this though, but I imagine it's maybe around the same lah. So I'm not really sure about any cheaper alternatives.
Project launches, how will it be maintained?
I don't know, man. If this project does happen, it's actually very easily maintained. Okay, so here's the thing. Okay, this thing, all you have to do is make sure the pipes are working and make sure there's like some UV lights to make sure that the water is clean when it goes in. And then like for cleanup, right, it's so easy because of the shape it is, right? That's why it looks like a menstrual cup lah. All the oyster shit or oyster feces will just travel down here into this black thing that can be collected by a boat. So it's very easily maintained. But as I've mentioned, for a cheaper alternative, right, if you don't want to do all this, at least farm them at a lower depth. That will drop the temperature of the oysters, the areas the oysters are farmed in, and that will also how to say drop the temperature, which will decrease the amount that dies and increase how much they can reproduce. But that's really not what farmers are looking for lah. They don't really care if the oysters can reproduce unless they are doing a hatchery. So it actually opens up another source of revenue actually for
Do you think
farmers.
it will affect the natural habitat in the areas?
Natural habitat. So for this one, closed system, some closed systems actually do, because for the like the fish waste and stuff like that, right, the feces, they just throw the water away back into the sea or back into the natural habitat. But for this one, we can collect all the rubbish inside the bottom here, and if we want to do water change, we can collect the water and then treat it and then introduce it back into the water, back into the sea. So if we take the proper precautions, it will not affect the habitat at all, or like hopefully. Yeah. Or what can be done, because if you all don't know about this, there's this thing called aquaponics. So aquaponics, basically we take the fish water, right, that has like feces and stuff like that, and we use it to grow plants because they want that nitrogen, they want that ammonia, that like nitrogen based ammonia and all those nitrogen compounds to grow. And then we can use these plants to actually clean the water and make more suitable water for the oysters. So we actually can recycle, or we can use the oyster water. We can collect the oyster water inside here if it's too dirty. Use it for hydroponics. Use it to for our Singapore plants that are, you know, in those greenhouses that are not in soil, that are just in like those rows of water. Yeah, I forgot what those are called. I think it's just hydroponics lah. Yeah, so we can use it for those. So we can just recycle whatever we use. And yeah, maybe if we like ship a certain amount of water every day to the farms, we can just keep this system going, and then put the clean water back in the sea, stuff like that. So this idea can be expanded upon a lot more, but for this is the rough idea of how farmers can actually solve the problem, just using this. But obviously the cheaper way of doing this,
You've been saying about collecting water, but how do we do the actual collection of it?
easy, we just use water pumps. Yeah, usually, yeah, we just use pumps and pipelines lah. Like even for our oysters, right, we actually use a siphon. Oops, siphon. Yeah, you've seen this before lah. We use this, and you can pump water without even like without even using electricity. We just press, and then the water just flows out by itself. But I guess for this one we can use electric pumps, so it's faster. It's a higher amount of water anyway. And electric pumps are very commonly used in aquaculture, so it's probably not a big it's probably not too much of a problem to do that. But obviously I'm not a farmer lah, so I don't really know everything about it. That's just from what I know how that can be done. Yeah, so instead of doing this, just farm them lower in the water where it's cooler, where they can grow better. And yeah, so that is basically it. And yeah, in conclusion, right, these oysters in Singapore are not being raised in the best environment that they can be, or like the optimal environment that they should be growing in, because Singapore's water is high temperature and high salinity. But we've seen that oysters grow best in low temperature and low salinity, because yeah, from all the results we've seen, right, like even though low temperature, low salinity is not the best in every single aspect, it's really not even bad results. Like yeah, for example, this one, yeah, let's just take this for example. Low temperature, low salinity, not the biggest, there's bigger ones, but it is the fattest. They have the most oyster, they have the least mortalities, they are able to make oyster with gonads, they have the least mortalities. What more could you want, right? So if you are able to somehow control the conditions inside where the oysters are growing in, maybe through semi-closed farms, lowering the nets to deeper levels, reduce what is it, the semi-closed idea I had, did I say semi-closed, or the closed farms, yeah, fully closed farms where we can do hatcheries and stuff like that. If we do that, we can actually reduce oyster mortality and also increase how much their reproductive organs can grow, or like how the quality of their reproductive organs and like their ability to reproduce will increase and skyrocket. And reducing oyster mortality, more survive, more for us to eat, able to meet the supply by 2030. However, when we have oysters that can reproduce in Singapore, it leads for a more self-sustainable food supply in Singapore itself. So as I've mentioned, Singapore is looking for the 30 by 30 campaign, right? 30% of our food supply by 2030. This is one of the ways that we can do that. So if it can increase our self-sustainability. We don't have to rely on imports. We have our own source of new oysters that we can even sell to overseas for extra money. And we also can increase Singapore's food security problem by, as I said, not relying on imports. So that is really what our project was about. It helped to increase the sustainability of Singapore and also find out that the rainfall itself affecting the salinity and temperature is not actually what affected the oysters or the oyster mortality. In fact, it actually probably helped them. You are free to you are free to think whatever. Others are free to think whatever they want to, but we found out that it's not these two. That it's not these two that is the issue. So others can speculate what else could be the problem. Yeah, so that is my oyster project. Anybody have any more questions? I'll just leave the floor open for that content, you know. I need to reach 10 minutes. Let me just close all this. Yeah. So if there is no questions, yeah, I assume there's no questions. Yeah, if there's no questions, I'll probably end the stream here. Thank you everybody for bearing with me and looking at my horrible drawings and hearing my explanation and being here helping me with my thing and, you know, contributing to the video and stuff like that. Yeah, thanks for coming. Bye bye.